Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Okay, hi everyone. We are really happy to have Matt Might here. Matt is from the University of Utah, but he is visiting Boston. He's visiting Harvard Medical School this year, and he is going to tell us a precision medicine algorithm. What do you do when you're one of a kind?
All right, thank you. Well, I appreciate those of you that had to brave the rain to get here. I'm glad you survived. As you can tell, my dress shoes did not. So I recognize this is probably a more computational audience than I typically speak to. I myself am a computer scientist, but I've been doing a lot of medicine lately. So let's just go over some of the basics first. So when I say something like precision medicine, what am I talking about? Well, you may or may not have seen an announcement from the president about the Precision Medicine Initiative. And this is really all about a big change that's happening in medicine. And I think there are two factors driving this change. The first one is a shift to exploiting genomic data so that we can actually use genomes to improve care and make it significantly more precise, as it were. And the other part of this change is the use of all the data available from patients to drive care as well. So, and in some sense, you can think of genome as just another kind of data, but it's a rather big kind of data with very special properties. So I think that's really the essence of precision medicine. And the way the president put it, he said this is all about getting the right drug to the right patient at the right time. That is the essence of precision medicine. And it's very strange for me to be talking about this because, again, I am actually a professor in computer science, and most of what I do is cybersecurity. So I always thought if I ever did give a talk at MSR, it would be on cybersecurity. Alas, I'm not here to talk about that, but maybe some other time. And the reason I got interested in medicine in the first place is that 3 years ago, my wife and I got some unbelievable news. Almost literally unbelievable. We were told that our son, it was in fact the first and at the time only case ever discovered of a brand new genetic disorder. So we had to start answering this question. What do you do if you're told that your child is literally the only case known of a brand new disease? So this is what we've been trying to answer for a little over 3 years now. In some sense, we've been trying to answer it longer than that because we had a long diagnostic odyssey beforehand. And the way I look at it, what we're really after is a process. We want a process that takes data in and maps it to a drug on the other side. That's really the goal of precision medicine in my view. And I think that's the reason computer science is so important in this process, because what we're really talking about is an algorithm that maps that data from the patient, their genome, their other health data, into that drug. And if you want the short version of what's involved in this process, what makes up the majority of it, surprisingly enough, I would actually have to say it's a matter of trying the internet. So it turns out there's an awful lot you can do with the internet these days. Maybe not so surprising to this crowd, but it certainly surprises older doctors. And the reason I think it's great that the internet can play such a major role is that it's like, hey, unless you live in North Korea, chances are good you have access to Google or Bing, as it were. So both of them work just as well. So what I'm going to tell you is, in some sense, the story of executing an algorithm on my son's case. And we'll sort of map out what that looks like by tracing the steps that we traced and showing you some of the branches that applies to other disorders as well. And I'm going to clarify that even though I'm talking about my son Bertrand, this is in no way a unique story anymore. In genome sequencing is discovering disorders at an unparalleled rate in human history. We're talking about hundreds of new diseases discovered per year, a rate that's actually accelerating at the moment. So, the input to the algorithm, of course, is a patient. And in this case, in rare disease, you end up with, again, people use the term diagnostic odyssey for a reason. And it often begins on a place that a lot of us call undiagnosed island, which is a place where you have a population of one patient or one family. And these odysseys take a varying amount of time, but they share some stuff in common. Now, in our case, we didn't know why my son was having seizures, or why he was developmentally delayed, or why he had a strange movement disorder, or why for some reason, and this is one of the standout symptoms, he couldn't cry liquid tears. So there's a lot of stuff that was very unusual about my son, and we just had no idea what was going on. But it's also an island where, as a parent, when you watch your kids go through this, you learn about this tremendous resilience within the human spirit and of the power of just never giving up and never giving in. And really, that was our life for quite some time. But I would say that even before we had a diagnosis, we were already engaged in what you might call precision medicine because we were collecting data and acting on data. Actually, back before you could even build iPhone apps, I built an iPhone app to track all of the seizures my son was having, along with all of his other medically relevant events, like medicine, like when he was getting it, how much he was getting, and all of his food. And by mining this data, we could figure out that his seizures were actually correlated to the timing and dosage of his particular medications. And by spreading them out, we actually eliminated about half his seizures from that. So even without a genome, you can do data-driven medicine. And of course, we actually did some other stuff that's considered part of the precision medicine initiative back then. Right now, the state of the art is you still get mountains and mountains of paper records when you're dealing with anything like this. And we were, I would say, religious about digitizing these. So I would take them into the high-speed scanner at work every week and add to the collection, and then end up posting them in a secure site online. So that every time we went to a new hospital or saw a new physician, my son had one of the very first truly portable EHRs, because we could send him a URL and say, here's your login, here's all of his records. And we OCR'd them so that they were searchable, so that they could search for anything, anywhere in his records. And this tremendously, I think, accelerated what we were doing with that diagnostic odyssey. And unfortunately, I wish I could say that we're better than this, but we're not much better than this right now. If you are spread across multiple institutions, this is more or less state of the art for EHRs. And that's just an embarrassment. So for 48 months, that was our life. We had no idea what was going on. And then we engaged in sort of what I would say is the first step of the precision medicine algorithm, which is sequencing. And a team of scientists from Duke University who'd been working for quite some time said, you know what, we think your son is a really good candidate for this new technology called exome sequencing. So that's what they did. They sequenced myself, my wife, and my son. And they used this so they could check for novel mutations and any ultra-rare inherited mutations. And what they found was he had, in fact, inherited a premature stop from me and a frame shift from my wife that led to loss of function in this one particular gene called NGLY1 or NGLY1. So this gene was knocked out. And that's when they also said at the same time that, hey, but he's the first and only case we have ever seen with anybody missing function in this gene. There's literally no one in the literature with a disease associated with this gene. So this is really our best guess that this is what's causing it. And they were pretty confident. But to raise confidence, you have to go to the bench and do some more stuff. So this leads to a very critical phase of the algorithm, which is interpretation. And a lot of this right now is done at the bench. And I guarantee you most of this needs to move in silico. There's a huge role for computation and interpreting variants. And to the extent it's being done in silico at all right now, a lot of it's being done with machine learning, although I think there's a lot of room for doing some rational construction of algorithms for variant interpretation. So what they did is they made sure that the protein wasn't there, and that's what this means. The protein wasn't there at all. It really did destroy the protein. But then what we had to do is we had to take my son and as a collection of data points, to take his phenotype and try to map it back onto this purported genotype. And to do that, you have to say, well, what does this protein actually do? Or what does this gene actually do when it becomes a protein? So we looked at the structure of the protein, and there's actually some information on the protein itself out there, and we knew it was responsible for deglycosylating misfolded glycoproteins after they're retrotranslocated from the endoplasmic reticulum into the cytoplasm. So, there are phrases you think you're going to say as a parent someday, and I guarantee you that was not one of them back when my son was being born. Anyway, it's a recycling protein, more or less. And if it's gone, then you don't recycle certain waste products inside the cell. So we said, well, if this protein's gone, we should expect to see an accumulation of these waste products, and this could plausibly explain the disorder. So to go further than that, we had to reach out to an expert in disorders of glycosylation, Dr. Hudson Freeze at the Sanford Brennan Research Institute, and we said, hey, if you saw a patient that had this defect, do you think it would cause this disorder? And he said, you know what, I think it actually would. I think you've got a brand new disorder in this case. So again, at that point, we were pretty sure that we had an answer to this disorder. You also oftentimes hear in instances like this that this is not an actionable diagnosis, that there's nothing you can do once you're told that your child is the first in the or even if it's an ultra-rare disease, they often say there's nothing you can do because there's obviously no drug companies working on a disease that was discovered yesterday. That just doesn't happen. So they were a little, and they even said it was going to take probably 5 to 10 years to find another patient because that's typically how long it takes when you get a brand new disorder like that. So But what I would say is that if there's one thing I could get across today, it would be that this phrase, which is a clinical phrase used all the time, it has really lost its usefulness and certainly lost its truthfulness when it comes to modern medicine. Starting with the easy stuff. So here's my third child, Winston, and the reason he exists safely is because we got an actionable, what I would say, an actual diagnosis. We were actually able to make sure that he would be born without this particular mutation, and that's something you can do if you get that information. So that's the low-hanging fruit for action. Because once you get an answer, you can start doing science. And this is the part that I think is going to have to scale up tremendously for precision medicine to work. And I often argue that in the absence of a medication that you can take, it's science itself ends up becoming the medication. It becomes the medicine, because science is a form of action. It may not be traditionally what you do when you get a diagnosis, but it's a perfectly viable thing to do in precision medicine. And I can tell you as a parent that as soon as your set of potential diseases goes down from dozens to one, then once you have one target to shoot at, you just go start looking for arrows. It's what you do. So that's what we did as parents. We started looking for arrows. And I think this is a process that really begins by checking to see whether or not you can build off of stuff that other people have done. So we checked PubMed, and we found out that there were, we knew almost nothing about this particular protein. So I did this search recently, and there's now 11 papers on this gene. When you did this search 3 years ago when the disorder was discovered, I think there was like 3 or 4. And this gene had been discovered 20-some years prior. So if you think about it, we've almost tripled the amount of our knowledge on this gene in just 3 years compared to what we had done in the prior 20. So there's actually, as it turns out, tremendous value in having a human model of a genetic disorder. You can learn a lot from them. So we realized if we're going to stand on the shoulders of giants, in this case, we needed a lot more giants. We need to start engaging the scientists early on. So we went right out to Dr. Hudson Freese's lab. We said, hey, let's work together. And we said, where have we arrived? What have we reached? And so I think in a conversation in a lab in one afternoon, he really took us to the peak of our diagnostic odyssey and showed us just how far we had come. And ultimately, how much further we would have to go if we wanted to reach understanding, treatment, and cure. And it turns out that we just done the easy part. Getting an answer is like the easy part of precision medicine, because now you have to go climb Everest if you actually want to do something about it. And that's the part we have to scale up. Now, I think the amazing thing is that we took our first scientifically plausible attack on the disorder only weeks after discovering it. So what we did is we looked at the mutations themselves and we said, is there anything actionable we can do on these 2 mutations? Is there something that we could use to influence them? It turns out there's an approach that works in about 1/3 of all genetic disorders, at least in theory. And this is where I learned that theory in biology is very different from theory in computer science. So in theory, there are these things called read-through compounds, where if what you have is a premature stop in your DNA. And you can think of a gene as just a long sequence of instructions that tells you how to build a protein. And a premature stop mutation just throws a stop sign in there early on. So it's like a halt instruction comes too early, halfway through the protein. But there are these read-through compounds, which at least in theory, can force a read through that stop instruction and allow complete construction of the protein. But when we tried all of the existing ones in my son's cells and dishes, none of them actually worked. So that's when we learned that the theory and practice differ quite a bit. And you might think it's discouraging to find out that none of these work, but I was incredibly invigorated to think that we could go from not knowing what this thing was a few weeks prior to actually trying out compounds in a dish. I was like, wow, maybe we can move faster than I thought. So I was very energized by that. But I also realized there's no way we could do this alone. We were going to have to actually find others and work together with them if we were going to solve this disease. And that's another really important step in this algorithm, which is to build a community. And to do this, we actually started looking in genomic databases. So you have these databases like right now down the street at the Broad, where you can look up a gene and ask, what are the frequency for pathogenic alleles in there? And you can use that to solve for what the estimated patient population size ought to be. And I came up with an estimate of about 500 patients out there somewhere in the world that were alive suffering from this very same disorder. So it's a 1 in millions disorder, but it's enough that there should be others, and we should be able to find them. And so I think coming home from the hospital that night, I said, I want to go find the others, and we hatched a plan to do exactly that. So what I did to find them is I used social media. I wrote a blog post that was designed to find other patients. And to do this, I engineered it to do 2 things. It had to go viral, and it had to rank very highly in Google or Bing, so that if other patients were out there searching for the same thing that they would find us. And that's exactly what it did. So this is the post I wrote. So this is the part that makes it go viral. And there's 5,000 words below that make it rank highly in Google. So that's, it's, I hate to say we exploited internet cat picture theory, but we did. The internet is like soberingly predictable sometimes. So yeah, this thing took off. I mean, it just rocketed out there. It was seen by millions in a very short amount of time, and ended up about a year and a half ago in the New Yorker. So this thing has just kept snowballing and getting ever more coverage. And it did, in fact, rank highly inside Google, so that if somebody was out there searching for the same symptoms, they would land on it. And then patients started showing up. So for a disease that no one knew existed, 2 months later, suddenly had a community, because we had 2 more patients in Turkey. And then a few months later, 2 more patients in Israel. And then a few months after that, we had a patient in Palo Alto. And over the past 3 years, patients have been popping up everywhere. And it's almost impossible to really describe what this means to an individual patient or to us as a family to suddenly have this community. But what I would say is it really washes away that dark night of uncertainty and isolation that comes with not having a diagnosis, and replaces it with the light of science and community. And that community is, in fact, very united. So there's now 39 confirmed patients, with several more likely to be confirmed in the very near future, that are all working together to try to figure out how to understand, treat, and cure this disorder. And now we can ask questions like, well, what can you do with a community of 39 patients that you just couldn't do when you only had 1? Turns out there's a lot. So as soon as you start having a patient community, nature starts running experiments for you. And if you're keeping along, this is step 3B.A in the algorithm. I won't keep track of them after this, but I'll mark where individual steps are, and I'll show you a flow chart at the end. So here's some example of natural experiments that happen. So this child right here, in addition to having NGLY1 deficiency, the same disease as my son, also had horrible liver cancer by the age of 1, which necessitated a full liver transplant. And the crazy thing is after his transplant, he recovered from more than his liver cancer. He also recovered from many of the symptoms of NGLY1. So this has sparked a number of hypotheses about what exactly might have caused this to happen, and it's definitely driving the clinically oriented science of this disorder. You also end up with patients like these 2 right here, where most of the patients are clustered in terms of severity down near the bottom end of the severe range, where these 2, even though they clearly have the same disorder, are extreme outliers. They're much more high functioning than all the rest. And so by volunteering their genomic data and their health data, they're helping us sort of crack the code of what makes them different. It might provide us targets that we can act on therapeutically. You can hold conferences if you have a community. So I think barely a year after we discovered the disorder, we gathered all the patients, parents, and scientists in one place and said, let's figure out what the strategy for going after this disorder is. And we're about to have our 5th conference in 3.5 years coming up in December. So we've made a lot of progress in terms of getting scientists to actually figure out what the key problems to attack are. You can build foundations. In fact, we have 2 foundations for our patient community. So I like to say that if we could maintain this patient-to-foundation ratio going forward, I think we're going to do quite well. But I think both foundations work very closely together to make sure that we're maximizing the ability to use our resources. And 13 patients have already gone through a natural history study sponsored by the NIH. So if you're not familiar with what a natural history study is, I'll show it to you in just a second. But I would say what this really is this is deep phenotyping. This is really uncovering the essence of the disorder at a very broad level. So I'm very grateful to people like Lynn Wolf, the study coordinator, and Bill Gall at NIH for actually supporting this research and allowing us to get 13 patients through in 1 year and then having them come back every year after that. So to give you a sense of what it's like to go through a 1-week, high-level NIH study, what I have here is a single picture for every poke, prod, procedure, or appointment that occurs during the course of this entire week. So in 1 week, all of these procedures happen. This has now happened to 13 patients. My son has already been through for his 2nd year. So if you do this on a large patient population, and 13 is getting large, then what you get when you aggregate the data are biomarkers, clear clinical indicators of what is happening in a disorder like this. And what we found, it was, I guess, less interesting to a computer science audience, but we found things like certain kinds of low protein in the CSF, and very unexpectedly hypoglycosylation in the CSF as well. So these sorts of things are very much guiding the basic science going forward, and also providing markers that an agency like the FDA could look at when you're doing clinical trials. But more importantly, when you start to get all this data, it starts to make that target bigger. It makes it easier to hit every time you uncover some chain in the mechanism of harm for the disorder. And that gets you back on a more traditional route of drug development. And one of the mainstays of drug development is something called screening. So it sounds crazy, but what I'm about to show you is really what state of the art is when it comes to drug development. What you do is you need to build an assay which can detect whether or not some compound has the desired effect. In this case, to build the assay, all we had to do was Google for it, because we were actually in the process of building it independently when my wife said, wait a minute, what if somebody else has already done this? And sure enough, there's already an assay out there for this particular gene. So we sent that over to Dr. Hudson Freeze, and he started working with it. And what you're seeing here is that the green glow means that the activity of the enzyme is present, and if you don't see green glow, it means the activity is not there. So if you find a compound that actually restores the activity, then you expect to see green. So what we're after with this is green. And what we really want to see is as much green as possible if you do this at scale. So there's these robots out there now where you can test literally millions of different compounds to see if any of them can give you any green glow at all. So we're working with a facility to do this at Sanford Burnham. And again, the idea here is it's very brute force. You just take as many compounds as you can get and you just try them all. And so you're not thinking rationally, which is what you might think you might do if you're doing drug design. And there is something called rational drug design, but that's not really all that popular yet. You just throw everything you've got at it and hope something lights it up. These days, you can also do a lot with model organisms, and the cost of model organisms just fell by probably 2 orders of magnitude thanks to a technology called CRISPR. You've probably heard of CRISPR, because you can very rapidly edit the genome of other organisms at this point. So we have a sort of like little island of Dr. Moreau of N-gly1 animals at this point. We have mice, we have flies, we have zebrafish, we have worms, and we have yeast. And I don't know if ours retain the wonderful nutty flavor of regular yeast. I'll try them out at some point, I guess. And you can even think of really large molecule therapies too, actual biologics. So in this case, there's a natural biologic, which is the enzyme itself. Like if we can synthesize the enzyme and just put the enzyme back in the cell, presumably that would give you the activity back. But again, this is a little bit easier said than done. And the first step is synthesizing this at scale for a reasonable price. Because it turns out the synthesis problem is solved. Because if you Google for synthetic human N-gly1, it's out there. You can just buy it on the internet. The only problem is if you look very carefully, it costs $402 for 10 micrograms. So again, if you just extrapolate that out to what you would need for a human patient, it's about $2 million a week. So it turns out most insurance companies will not go for this. So we're going to have to work with the insurance companies on whether or not this is a reasonable strategy. But even if you do that, it turns out there's still some biological problems that stand in the way, like the fact that if you just inject this enzyme, it actually doesn't go to the right part of the cell. It ends up in something called the lysosome when it needs to go to the cytoplasm. So first you have to pegylate it for a number of other reasons. And then you want to tag it with a peptide stolen from HIV to make it cell penetrant. So this is enzyme level engineering. And if you do this, then it gets into the cytoplasm, then it might actually be biologically active in the right place. And of course, we're also very focused on repurposing. And the reason I like repurposing is that it's a very computational way to go about drug development or drug discovery. And the whole idea here is that what you have a coordinate who's essentially some sort of deviation in what I'll call phenospace, the space of phenotypes, where each of these is some feature that's out of whack. And what you're looking for when you do drug development or repurposing is the drug or the combination of drugs, really, that is the exact opposite of the patient in phenospace. So that when you bring these two things together, what you've done is you've canceled out all the features. And there's lots of ways to slice phenospace. So you can look at RNA expression, you can look at metabolics. There's lots of ways to look at what is happening to a patient and the effect that a drug might have. And again, if you think about it in some sense, what you have now is an optimization problem. You want to figure out what combination of drugs gets you closest to reversing the effects on the patient. So in fact, at Harvard, there's projects going on right now in the group I'm with for figuring out how to compute drug cocktails that will exactly reverse a phenotype. And then I think another really cool approach you can take, building on some of the earlier efforts we did with social media, is crowd screening. So I'll talk now a little bit about the potential here. So the crowd screening, I think it can be as unstructured as just sort of blasting stuff out to social media and asking people what they would do, to the much more structured where you actually engage in some of these citizen science initiatives. But some of the most interesting suggestions we've had have actually started on social media. So by broadcasting over Twitter, we ended up connected to a postdoc in Texas who said, hey, I'm working on cystic fibrosis, but I actually happen to need cells that look like your son's cells, because he happens to have the mutation I care about right now. So we shipped him some cells, and what he reported back a few weeks later was that with his new compound he's working on for CF, it's a read-through compound like I mentioned earlier, he was actually able to restore about, and it's really hard to see here, but about 1 to 2% in normal levels of NGLI1, which is probably enough to actually be biologically viable at that point. So we're, again, a connection over Twitter has led to a pretty promising lead in terms of a novel way of going about read-through. And we've been working very closely with him ever since to see if we can actually go to the FDA. So actually, that's the phase we're in right now. We're trying to figure out, with the FDA, what would it take to start a trial based on this compound. There's a company right here in Boston that's actually feels the science of NGLI1 is now promising enough, or sort of, I guess, sufficiently de-risked that they're even interested in starting up a unit to do drug discovery for NGLI1. In fact, they have started that unit for NGLI1. Based around a different approach to therapeutics. And we actually have a much more structured citizen science or crowd screening effort going on with Andrew Su at Scripps, because he has a platform called Mark to Cure. And what Mark to Cure does is it allows patients, or really not patients, but any volunteer that wants to, the opportunity to mark up abstracts so that they can highlight what the genes are, what the diseases are, and what potential therapies are. And if you do this over enough, abstracts in PubMed with enough volunteers. In this case, this graph was generated, I think, with several 100,000 annotated abstracts. You can start to draw out connections in the literature between disorders and treatments. And a very interesting path that popped out literally last week. So we had a conference call last week where I saw this graph for the first time. And they said, does anything on this graph look interesting to you? And I said, there's definitely one very interesting thing on this graph to me, because one of the most successful prior treatments we'd ever attempted was on this graph. And there's even a compelling rationale for why it's there. So it connected NGLI1 all the way through a phenotype for a completely different disorder, but very similar disorder, in fact, through the lack of tears, to ACTH receptors and to ACTH. And ACTH has it's been used on several NGLI1 patients now, has a pretty remarkable effect. But the weird thing is it doesn't affect tears. What it affects is their seizures. So after about 2 months on ACTH, the seizures in NGLI1 patients go from more or less permanent background epileptoid activity to none at all. Their seizures are actually totally gone after 2 months on ACTH. And at the patient level, something really remarkable happens. So when my son went on this, he actually started smiling and laughing for the first time. So prior to this, his life was pretty awful, but in fact, he's been laughing ever since he's been on it. It was a very permanent change once we tried this treatment. So ACTH, which showed up in the search, was actually sort of the key to unlocking his humanity, and certainly, I would say, his sense of humor. So yeah, I think the fact that it showed up there was really incredible. And the cool thing is we never knew why ACTH worked. But here, there's a compelling explanation in terms of its connection through the phenotype, but also a family of receptors over here that we're looking at now.
If you didn't know why it worked, why did you give it to him?
Because sometimes it works in intractable epilepsies. So it's like literally the last thing you will ever try in an intractable epilepsy, because it will almost kill the patient. So no one can be on it long term. That's the problem with ACTH. So my son ended up in the hospital for 3 weeks after 2 months. But it did permanently eliminate some of his seizures, and then some of them came back. But we never understood why. But now we have a whole family of receptors to look at that interact with ACTH, and hopefully we can target the right ones and figure out the ones that are actually suppressing the seizures versus his immune system, because that's the problem. It'll suppress your immune system. In fact, just wipe it out entirely. So that's where we are now. So we're doing another deep dive with this same citizen science initiative, where we're focusing on just the literature surrounding here to see if we can figure out what the mechanism for seizure suppression actually is. And I'm optimistic that we'll actually turn something up. And another sort of key feature of citizen science is what they're calling patient-driven science, or precision medicine. It's really this patient-driven science. And I myself have actually spent a fair amount of time switching from we're really not switching, just really adding another aspect to my research portfolio in the middle of biology and medicine, and particularly the intersection of biology, medicine, and computation. And I've spent more or less 3 years getting up to speed, and I'm now so up to speed on this stuff that I was actually able to get a grant funded for doing drug development for NGLI1. So over the summer, we actually built a colony of planarian worms that have my son's disorder. And the nice thing about planarian worms is that if you cut them in half, you now have 2 planarian worms. So if you want to grow up a large colony quickly, once you've got one, you have many. So that's a nice feature of this particular model organism. And it turns out this worm can get a seizure too. So that also makes it a great platform for testing drugs. And people always ask, can you replicate this? Can you actually scale this process up? And the answer is absolutely yes, you can. You can definitely scale this up. But I know you can do this because I've seen it done in other disorders now. I have a lot of patients that reach out to me and say, we want to start doing the same thing. And so I've helped a number of disorders start taking these exact same steps. This is one of the first ones I helped a few years ago. And they said, what we were told, we did genome sequencing. They said, well, it could be this mutation, could be this mutation, could be neither of them. We don't actually know. We need to find another patient. So I told them how to put together a website that would actually help find other patients. And 29 days later, they had another patient. And it confirmed that it was, in fact, one of the variants that was flagged by sequencing. So if you think about that for a second, that means, in this case, it was 2 moms exchanging emails who ended up on the front line of science. They were the first ones to actually know that this was a brand new disorder. And this is something that's going to happen over and over again, where the patients are just thrust out to the frontiers of science and end up being the first ones to know something instead of the scientists. Another mom who said, well, I like the idea of using the internet. I already have a blog. She after she read our story, she did it herself without contacting me, which I think is great. You don't have to contact me to put your kid's case on the internet. And within, I think, a couple weeks, she had 8 cases of a brand new disorder. And then just a few weeks ago, while I was here over the summer, I was working with a family that said, we also need to find another case for our disorder. And here's the incredible part. So if you just look over time at the decrease in time it's taken, when they put their post online, the time it took to find another case was 12 hours. 12 hours. So it took us 2 months, then 29 days, then a few weeks, then 12 hours. So the amount of time it's taking to find other patients online is dropping very rapidly. And of course, we're getting better at it too. We're getting better at exploiting social media to do exactly this. And the reason this is so effective is that there's no one large database of all variants out there. But the internet itself acts as a kind of database already. So they were able to get through to researchers who were sitting on unpublished data about that particular gene and who had, in fact, knew other patients. Yep.
Would there be some benefit in making that process more standardized and connected? So anybody who got a sort of like, we have no diagnosis for you, creates a profile and makes themselves known, but in a way where like your son's profile sits next to that kid's profile, and then the researchers would look across that.
That is exactly my next slide. Perfect question, perfect timing. Yes, there's tremendous value in standardizing this process, and so the NIH has just backed a major effort called the Undiagnosed Diseases Network, part of which is headquartered at Harvard in the group I'm with, to scale this process up. So they're going to do that week-long deep phenotyping, and through a partnership I have with Kim Splinter and Rachel Ramoni over there, we'll actually be putting these patient profiles on the web in a very systematic way. But I feel like one thing I need to mention is that one of the most important things you can do is to put the information on Wikipedia as well. There's some strategic reasons for that. So if you're not familiar with Wikipedia, because I suppose you've been in cryostasis for 15 years, we have a lot to catch up on. But let's start here. So it's become the world's database. So there is a default database for everything now, and it is Wikipedia. And obviously anybody can view it, anybody can edit it, and this makes a huge difference. And patients and parents search this all the time. I didn't know anything about biology prior to all this. Everything I know I learned from Wikipedia. I took 6 weeks of really crappy biology in high school, and that was it. So it's been Wikipedia for everything else. And the nice thing about Wikipedia is it's naturally self-optimizing. It naturally rises to the top of search engine results. So if you put information there, there's a good chance people will find it. So I even created a blog post that tells patients how to do this and all the other stuff I've been recommending. And I even have a Wikipedia entry generator. So you can just type in the information and get out exactly the text you need to copy and paste in Wikipedia for your particular disorder. So this makes it very easy for any patient to get their data on there and connected to other patients as a result. They can just add a section called clinical significance, whether it exists or not. The other thing you can do which of course applies to Bing too, is you can buy AdWords. If you don't want to take the time to go edit a Wikipedia entry or get your post to go viral, you can just pay Google to rank at the top, and we do this too. So I think if we really do want to scale this entire process up, from patient finding all the way to disease discovery, then we do need to pin down what this algorithm looks like. So I'll describe it now sort of at the level of a flow chart that takes you through this entire process. So again, what comes in as the input is an undiagnosed patient or a patient seeking information. And I think the next step in precision medicine is almost invariably going to be sequencing. You're going to have to figure out what is going on in their genome. But this then involves actually interpreting the genome, which is, again, quite tricky because you have to talk to gene experts, you have to do maybe studies in a lab, and also, I would argue, turn to the web to see if the web happens to know what is going wrong with any genes of interest that pop up. So eventually, you get an interpretation. You say, well, we think this is the gene. That's problematic. Or in the case of cancer, you'll probably end up with multiple genes that could be driving the cancer. But you get some genes that are acting to cause a pathology. And then, depending on the function of that gene and what has happened to the function of that gene, there's other stuff that starts to happen. So you can get a gain of function. Maybe the gene is now overactive, or a partial loss of function where its activity is reduced but not gone, or a total loss of function, like what you have in my son's case. Or, in the worst case, I think what you have is a change of function, where now the gene went from doing one thing and now it is a completely different thing. That's harder to deal with, but you can still deal with that. And the other thing you can do is, of course, you can start doing the deep phenotyping, and you can move into crowd screening as well, regardless of what has happened to the gene itself. So I'll just talk now about some of the paths down these branches to show you what they look like. So for total loss of function, there's a lot of stuff you can do. And some of this stuff also applies for the other branches. But you can start building model organisms. You can do structural analysis of the enzyme itself or the protein itself. It's not always an enzyme. And you can even start developing assays. And once you do these things, there's lots of other stuff you can do as well. So I'm not going to go into all these in detail, but I'll talk about some of the low-hanging fruit, because I think the low-hanging fruit is actually really important to try at scale. So one of the lowest hanging fruits for total loss of function, particularly in a metabolic disorder like my son's, is to try a metabolic diet. And here's how you go about figuring out what a metabolic looks like for your particular disorder. So if you think of an enzyme as a function that takes in A's and gives you back B's, then a metabolic disorder is what happens when that function's gone. And you can no longer turn A's into B's. And what this tends to result in is a toxic accumulation of A's and a deficiency in B's. So then there's a very simple logic that follows, which says, okay, if that enzyme is gone, can you give B or can you restrict A? That's a metabolic diet at that point. And it's localized to the specific enzyme that's been knocked out. And there's plenty of examples of metabolic disorders where this actually works. So for PKU, if you can restrict phenylalanine, it's more or less a cure. And then for CDG1B or CDG-MPI, if you can supplement with mannose, again, it is more or less a cure for the disorder. So then, of course, you have to ask, well, what does this do in the case of NGLY1? It's also a metabolic disorder. So to do this, you actually have to go a little bit deeper into the basic science, because it's not, in some sense, what the output of the process here is waste, or what we thought was waste. And what we were able to do by looking at the basic cell biology of the disorder was realize that in the absence of this enzyme, something particularly interesting is going on. When this enzyme is gone, you're not just getting these free-floating glycoproteins. What's actually happening is this very structured form of protein aggregation. And this protein aggregation is actually, tripping up the recycling mechanisms that what you end up with is a deficiency in one particular rare monosaccharide. And that rare monosaccharide is represented in this diagram by a blue square. And it turns out this little blue square is available for purchase on the internet. It costs around 50 cents a day if you're talking about therapeutic quantities. So realizing that, I went ahead and ordered some. I did a clinical trial on myself for about a month. I didn't die, so I realized it's probably safe. So, in my household, I am the FDA. That's basically how it works. And so when I was pretty sure it was safe. So that's a great question. So there's several other disorders where the treatment involves these unusual monosaccharides. And so I took my best guess based on the quantities and the bioavailability of these other monosaccharides to try to figure out what would be reasonable in my son's disorder. And that's all I could do.
But you just took your wide weight, divided it by his, and then multiplied.
Yeah, exactly. Yeah, so we standardize on a dosage per kilogram. But yeah, I mean, it's ultimately, for any drug, you just have to sort of do this. Usually you do it with more people and not yourself. But that's what you have to do. But the amazing thing is, about 3 days after I gave this to him, he actually cried his very first tear. And actually started crying plenty of tears after that. So when we saw that first tear, it was, I would say, a pretty amazing moment. And what I did is I actually collected the tear, put it on dry ice, and shipped it to a lab in California for analysis, which is what any parent would do in that situation. I'm sure you all did the same thing with your children's first tear. And so, maybe it may have been one small tear for Bertrand, but I think it's really become sort of an ocean of science for the larger disorder. Because what it did is it said, okay, there is a deficiency component to this disorder. There's also an accumulation component, which is much harder to deal with. But there was something else out there that was driving the harm as well. Now, so that's loss of function, and that's one part of loss of function. You can also end up with gain of function, and here you want to look at other strategies, like can you stop the mutant protein from being produced or prolifically produced? Can you find an inhibitor that will block the activity of this overactive gene? Can you build model organisms, or can you also look at dietary changes? So in the same way that you might want to, but here the logic gets a little bit inverted. And then the important thing here is that sometimes inhibitors are easier to build than you might think. So there was one family I was working with about a year ago where they got their diagnosis 2 years prior, and it was in fact a de novo gain of function in a particular gene. And at this point, the next reasonable step to me, and I would have thought to any physician, would be to at least Google for whether or not there happens to be an inhibitor for this particular gene. Makes sense, right? Well, no one had done that, so I did it, and sure enough, there's multiple inhibitors for this gene that were available. Now, nobody knows if they're safe. Nobody knows if you can actually use them, but it's a starting point. You could do medicinal chemistry on this if it's not safe. So I think the moral of the story here is sometimes the obvious gets missed even by physicians and scientists, or maybe it's so obvious that they don't even think to tell a patient that that's what they should do. And that's why it's important to ultimately very standardize this process so that as soon as you see gain of function, the next thing you do is run this Google query. And if necessary, engage in medicinal chemistry to improve toxicology and make it more bioavailable. And then for partial loss of function, you sort of invert what you do for gain of function. You want to see if you can upregulate the one working copy of the gene, if there is such a working copy, and engage in many of the same behaviors. And for a change in function, then I think what you really want to do here is anything that ends in omics. So that's transcriptomics, metabolomics, proteomics, you name it. I think you want to do basically all of that, try to figure out what the heck has just changed with this particular protein. Because when it's a change of function, it's really hard to figure out exactly what's happening. And there's another less scientific step in this algorithm, which I think is really important for all patients and really all stakeholders to be engaged in, which is advocacy. So I spent a lot of time engaged with policymakers trying to explain to them that our current regulatory infrastructure is totally unprepared for precision medicine. And that in some ways the science is a lot easier. And I'm not saying science is easy, but in some ways it's a lot easier than the policy changes that are going to be required to make this really lift off. So I'll give you some examples of that. So my wife has been very active in advocacy back in her home state of Utah, and actually got she testified before the state house and the state senate with my son to get the right to try law passed. This is a law which was starting to propagate to many states now. And what this does is it says if you have a terminal disorder, then you have a right to try anything past phase 1 FDA safety testing. So if it's safe, then you have a right to try it, whether or not it's been proven effective. And odds are it's not going to ever be proven effective for our particular disorder anyway. So safety is all we're really after. So that's now on the books in Utah. And my son was certainly very happy to be a part of this process, mostly because he got the pen afterwards. And I spent a fair amount of time myself working on Capitol Hill trying to educate congressmen and senators. So I've worked with Diana DeGette, who actually found our story through the New Yorker and then invited me to come speak with her to sort of give her my perspective on precision medicine. I met a few weeks ago with Orrin Hatch to talk to him about what's going on in the space and what kind of changes might be made. And I've even worked with some higher level folks at the White House on what changes are ultimately going to have to come about to make this a reality. And I'm optimistic that in the long run, we'll actually get there. I think there's been some really uplifting progress on this front in terms of recognizing the regulatory obstacles that are coming to make precision medicine scale. So, my parting thought for all of this is that if there's one thing I've done today, it's that I hope I've convinced you that not actionable simply doesn't belong in our medical lexicon anymore. It's just not true at this point. Because even in the event that there is no other course of action to take, it is always possible to do science. So, thank you. Yep.
Right to the start. And wow, your visuals. I don't even want to know how long you took figuring out how to make your arrows shoot the right things at the right time, but very impressive throughout. I'm wondering about, clearly it makes total sense to reach as many people as you can, and the internet is the obvious way to do that, and you've had great success with that. Thinking about the global scale, both of genomes and of internet use, there's obviously a lot of populations, and you end up with 35 out of what you estimated 500 cases, or 39. Are there, do you have strategies outside of the internet for trying to find those other people? How are you doing that?
We do actually. In fact, in terms of like most patients we've ever found in one day, our most successful strategy was to set up a booth. And this is all my wife's doing. She set up a booth at a major genetics conference. This big, colorful booth that said NGLY1, and we had my son there and some other patients there. And clinicians just walking by are like, oh, I have one of those patients. We're like, really? Well, why didn't you put them in touch with us? They're just floating out there in Norway or South Africa or South America, disconnected from the broader patient population. They said, well, they didn't have a novel mutation, so we didn't think there was any value in it. It's just, well, sure, there's no scientific value at that point, but there's certainly value to the patients themselves to be connected to the community. Yeah, so sometimes the physicians don't even check to see if there's a patient community out there, but just walking, having them able to walk by this booth at a scientific conference made a huge difference because we found 5 patients in one day. Yeah.
So a comment on that question. First of all, just phenomenal what you've gone through and what you've accomplished. You said that advocacy isn't the scientific part, but I would say, like, I don't know if you've read it, but the book Impure Science by Steve Epstein is a tale about how often advocacy is part of the science, and you've had to discover that on your own. You might find the book really interesting. It's about the AIDS crisis and the people who had to educate themselves about the science in order to go and testify and convince people to do drug tests. So, great book. What in the algorithm that you drew, where along the way is the part that, while it may have worked in your case, is going to be the hardest part in other cases? So I'm thinking either there's a part of that like is hugely expensive, or for certain kinds of diseases isn't so easy, or you don't have the research backing to do it, or you had it in one case and not another. So like, where in that flow is kind of the sticking point for this to happen?
Sure.
With other specific disorders.
So the part where things there's 2 places where things get very expensive. So basic science is expensive, and people are very reluctant to engage, and patients, I would say, are very reluctant to invest in basic science for disorder. We've invested a lot in basic science for the disorder. And I think that's because I myself am a basic scientist. I understand that you don't know what you're going to get out of basic science. It's a lotto ticket. You're just hoping that something will pop up. And inevitably stuff does, but you can't know what that will be until you just do it. So just sending people to the bench to look at cells under a microscope, to stain these things, and to figure out what is going on is incredibly important, but also very expensive. It's not cheap to hire postdocs to do this sort of stuff. So that's certainly one aspect of it. Another aspect where things start to get expensive is screening. As you start talking about millions of compounds going through a screen, you're now talking about millions of dollars for screening as well. So, I think in the grand scheme of things, millions of dollars is nothing compared to what they say is the 2.6 billion that it currently costs for drug development. And I think there's lots of ways to potentially short circuit some of the costs in drug development. So right now there's this very sort of well-defined model where you start off, where you get something working with cells, and then you scale it up to model organisms, and for model organisms, then you start to go to first in man, and that's a very expensive process. And clinical trials, which I'm now encountering, are also ridiculously expensive. clinical trials are just off the charts expensive. You know, I mean, we're talking about, you know, for each patient, hundreds of thousands of dollars per year in costs. And these where you need thousands of patients, and these trials can run for years. So clinical trials are incredibly expensive. And I think, you know, for clinical trials, there's a good regulatory solution. So I was actually out speaking at Medics this past weekend where I unveiled my strategy, which is we have to go back to decoupling safety and efficacy, particularly for these ultra-rare disorders where we're never going to have enough patients to get the kinds of statistical confidence that you're after in a randomized controlled trial. All we're ever going to have enough to get is safety data. And once we've got safety data, then you say, well, over time, we can do retrospective analysis and try to figure out whether or not it really was effective. And in most cases where the diseases are very chronic and typically monotonically decreasing in terms of their phenotype, if it has an effect, it's usually pretty obvious. You know, if you give this sugar to my son, he has tears. If you take it away, the tears are gone. You know, I don't know how many, you know, placebo-controlled trials we need to do to actually prove the FDA of that, but it's pretty obvious to us that it's working. You know, ditto for something like ACTH. You know, if you put them on, the seizures are gone. If you take them off, the seizures go away. So you know, I think ultimately what we need to do is incorporate a sense of logic to the process. So if something that is logically impossible for the disorder happens after going on the drug, then you know it really boosts the probability that it actually worked. But that's not, you know, logic is not part of the process right now. So that's you know, screening and the basic science and the regulation really ramp up the cost. Absolutely. Yep.
So I guess presumably the FDA's like rationale for this regulation is that you might feel like or might be pressured into trying these drugs that aren't proven effective and yet are very costly. And I don't know how you.
Yeah. So that's obviously a challenge. You know, so if you go to your doctor and say, well, there's a scientific rationale for trying this drug, but then a payer might say, well, show me the published paper that shows that in 25 patients this actually did something. You're like, well, we don't even have 25 patients for the disorder, so we're never going to have that. So there's definitely other conflicts coming. Like how do you convince payers to pay in the absence of evidence in literature, where all you have is something plausible in terms of the underlying mechanism. And for that, I think we're going to ultimately end up using what I'll call N-of-1 trials, where we figure out how to actually determine whether or not for a single patient a drug is actually having an effect. And this is now emerging as a very serious topic in the medical community. How do you actually determine efficacy on a single patient? And then the model shifts to a drug company will only get paid if the drug actually makes a difference. And if it doesn't, then they don't get paid. So I think it also pushes the incentive in the right direction for the drug companies as well, so that so they don't get paid no matter what. They only get paid if they actually help the individual patient. And then ultimately, I think what we're going to see is a tremendous amount of repurposing. Once we sort of get through these obstacles, we'll see you know, an enrichment of the mapping between chemical space and phenospace, where we know which drugs are making an impact on which aspects of every kind of phenotype. And it's, you know, I think very likely algorithms will be the ones deciding what the most appropriate medication to take is in the near future. Hopefully near future anyway. Yeah.
How much better do you think patients who have a known disease that has some maybe not so great treatment could do by following your...
Yeah, I mean, if you have a known disease, then what you do is you chop off the front part of the algorithm. You just skip right ahead to the diagnosis part. And you probably still want to do sequencing though, because you probably want to know the precise mutation. Because again, read-through compounds are getting a lot better, and for about a third of all genetic disorders, at least one of the alleles should be susceptible to a read-through therapy. So you can think about attacking, you know, the mutation itself for an individual patient. And there's other strategies for attacking mutations as well. There's stuff like exon skipping. So if the mutation occurs in a region of the protein which is non-essential, you can introduce a complementary RNA sequence which can actually cause you to skip over that part of the sequence when you actually build the protein. So there's a lot of strategies coming online that could be very patient and very mutation specific that could very well work for known diseases. And for multigenic disorders, which is I think is what most common diseases probably are, then you can certainly apply the same logic here, but obviously a little more scaled up. And you know, the same kind of thinking applies to cancer as well. But cancer is also highly multigenic. You have tons of mutations typically involved in cancer. So in some sense, the way I've been pitching it is rare disease, these rare monogenic disorders, they're the easy case for precision medicine because you have a single non-moving target. whereas with cancer, it's just constantly changing all the time. And you might have to compute an ever more complicated cocktail to get it under control. Yep.
You mentioned at the beginning patients who, like liver example, and other patients who were higher functioning or whatever. Yep. Has anyone looked at whether this is multigenetic?
Well, so in some sense, all disorders have a multigenic component. It's clear that the two outliers must have some modifier mutations so that are suppressing aspects of the phenotype. And we're going to try to find those. That's what we need to do. So what I'm trying to do right now is set up a bioinformatics project back at Utah where we take all the genomes of all the patients that have been through the deep phenotyping and try to correlate them with the phenotype we're getting out of this NIH study. And there's plenty of bioinformatics pipelines that can attempt to answer questions like that. The key thing here is that you need to get enough genomes so that you don't have tons of false positives. You really want to know what the actual modifiers are. It is. But with 13, I'm confident we're going to get a list of maybe 100 genes. Who knows? Yep.
Have you had any contact with the Pfizer Rare Disease Institute in London?
No, I haven't actually. I've had other interactions with Pfizer, but not with that group.
Yeah, they set up this group in London that's, it's probably bigger sample sizes than you're talking about, but they're talking about knocking out in hemophilia, fixing that problem. True, the treatment might cost $1 million, but over a lifetime of treatment for hemophilia, the present value of that investment might look very interesting. So
absolutely,
another modality of thinking.
Yeah,
it's an interesting group.
Yeah, no, you raise an interesting point, which is that rare diseases aren't cheap, even if you don't cure them. They're really expensive for many of the patients, and by extension to the insurance companies too. So the insurance companies may also be incentivized to invest in developing some of these treatments because it could actually make it could be worth their while. It could be profitable to cure a patient instead of paying for a lifetime of treatment. Yeah, that's if we can bring the cost down. If we can really scale this process to the point where that's reasonable. I think over the next decade that might actually happen. There's a lot like CRISPR, is a huge breakthrough in terms of reducing the cost of building model organisms and then as a result doing very precise drug screening on them.
Any other questions? Okay, well we're going to have some treats out there where you can ask Matt some questions, and then a group of us will get together and keep on talking with him about various ways of scaling this. And if I didn't invite you to join that group, but you would really like to join that group, please let me know.
Great, thank you.